Okay. So another example of differential staining, we have endospore staining. So endospore stain is a differential stain, of course, used to detect the presence and location of spores in bacterial cells. So take note, only few bacterial species can able to produce endospores. And among them, we have, of course, the Bacillus and Clostridium. Okay, so most members of Bacillus, of course, they are found in soil, freshwater, or marine habitats, and there are soil, freshwater, marine saprophytes. But few are pathogens. Okay, so example, of course, you have Bacillus anthracis, and that's the one that can cause anthrax. And another thing, we have Clostridium that are usually found in soil, and there are soil or aquatic saprophytes, and sometimes inhabitants of human intestine. And some of them can be pathogenic. So example, of course, you have Clostridium tetani, Clostridium botulinum, Clostridium perfringens, and etc. Okay. So of course, Clostridium tetani is the one that causes tetanus, and then Clostridium botulinum, the one that can cause botulism. Okay, and another thing, they can make this endosperm, and endosperm is actually dormant. Okay, and so um, what happened is the endospore allows bacteria to survive even in a harsh condition environment. And take note that endospores are actually heat resistant. Uh, yeah, they are resistant to heat and other chemicals, and actually the composition of endospore is almost same with our hair. Yeah, it's made up of keratin, and yeah, so it's a tough covering. So every time we put stain in the endosperm, it's so difficult. So that's why it's so hard to visualize it. So for endospore staining, we have this um, method we call Schaufer Fulton's spore stain or endospore staining. So usually they use malachite green as a staining agent. So anong meron with malachite green? So first we have spore producing cell and non-spore producer. And of course, they are transparent um, under microscope or if without stain. Okay, after staining with malachite green, of course, both cells appears green. Okay, and we use heat to force the stain into the spore. So take note, spore are kind of resistant to stain, so that's why we need to use heat to allow the dye or malachite green enters the spore. Okay. So after that, after heating, of course, we use um, another decolorist step, which is decolorization, sorry, um, to wash away malachite green. So, anong difference ng malachite green with other dyes? Um, Take note, malachite green is water soluble, so it can be removed easily by water. So, mabilis siyang ma-wash away yung malachite green. That's why it's very good for endospore staining. So, and since water cannot penetrate inside the um, endospore, so the dye will remain inside the endospore. So it will appear green, and of course, to visualize spore producer and non-spore producer under microscope. So after decolorization by water with water, of course, saponin nawala. So we use of course yung dye nawala. So we need to use a counter stain, safranin, and of course the color of safranin like red to pink. Okay. So, under microscope, yung parent or yung spore producer, um, color pink, and of course, we have this spore inside. Okay, so um, so it's a differential stain because we can differentiate which one is spore producer and non-spore producer, as well as we could determine the um, morphology, okay? I mean the shape of the endospore, whether it's spherical or elliptical. So in this case, it's spherical, okay? And what else? Another thing we could also differentiate, like evaluate the position of the endospore, whether it's like center or terminal or near terminal. Okay, so this is an example of endospore staining, and as you can see, um, uh, same bacteria, but they have different number of spores. So why? So take note, since spore allows the bacteria to survive in poor environment condition, we need to induce or to mimic that effect. So usually culture age can affect sporulation. So pag bata pa or young yung culture, usually hindi sila nag-form ng spore. Unlike pag matanda na yung culture, usually they and then nagdi-deplete na yung nutrient dun sa medium, usually the bacteria uh, undergoes sporulation. Okay. And then another thing, we could also differentiate the shape of the spore. So here we have Sporosarcina urae spore. So yung shape niya is actually spherical. Okay. And here we have elliptical spores. Okay. Ito ay Bacillus megatherium. Okay, so they have elliptical spores. Okay, and another thing here, like most spores are found like sa center, so therefore this endospore central elliptical. Okay, and then we also have subterminal. Okay, and like Clostridium botulinum for subterminal, and of course for Bacillus megatherium they have like central. Okay, and here we have elliptical terminal, so mapapansin nyo parang pregnant siya sa dulo, uh, may bulge or swelling, so this is Clostridium tetani. Okay.